Hey, what's up guys? My name is Alex Warren, and if you're here, you're a bum. It's the day before the final and you haven't studied. Don't worry though, you're in the right place because in the next however many minutes, I will be going over everything you need to know about genetics in Bio 182. So let's not waste any time and get straight into it. Okay, genetics. Genetics is the scientific study of genes, their functions, and heredity, which explains how traits are passed from parents to offspring through DNA. Pretty basic stuff, but it's important that I say it. Oh, and also, I have linked in the description a couple of the Quizlets that go over all the terms that you should have memorized for the test. And that's not just this unit, that's all three. Okay, so back to genetics. We start with Gregor Mendel. He was a learner, teacher, scientist, and man of faith, but of course we know him for his contribution of his pea plant experiments. In these experiments, he took true breeding pea plants, bred them together, and measured their phenotypes and their ratios. It's a lot to take in, so let's break it down. True breeding means that the parental generation, or the pea generation, the original plants where all the offspring got their genes from, were homozygous for their respective alleles. Allele being different forms of a gene. So they were either homozygous dominant or recessive. Phenotype is the physical appearance of the trait. You can remember this because genotype has gene in it, so you should be thinking about chromosomes, alleles, and kind of the abstract code for the trait. And so phenotype must be the physical aspect of the trait. We'll come back to the ratio part when we get to Punnett squares. Okay, so let's talk about the outline of his experiment. He had 3 generations of plants. The original group that I initially talked about, known as the pea generation. And remember, this generation is made up of homozygous individuals. So after breeding different genotypes of the pea generation together, also known as hybridization, you get the F1 generation. This generation is made up of only heterozygotes. We would call these individuals heterozygous individuals because they have one dominant allele and one recessive. Now, how did this happen? In order to understand that, we must look at a first Punnett square. So what are Punnett squares? Well, a Punnett square is a genetic tool that uses a diagram to predict the possible outcomes of alleles during breeding experiments, showing the probability of an offspring inheriting a particular genotype and phenotype. Okay, so how do they work? Most of the genes in this class that we focus on only have 2 alleles, and when these 2 alleles come together in different combinations, you get your genotype. Mendelian genetics states that one allele is always dominant to the other one, which means that in these heterozygous combinations, the dominant allele will always be the one shown in the organism's phenotype. How Punnett squares work is that you take each of the parent's alleles, line them up in a table like this, and then you kind of cross multiply them to find the possible genotypes of their offspring. When we do that for the pea generation, we see that their alleles cross multiply in a way that all of their offspring have the same heterozygous genotype. When Gregor Mendel did this cross again with the F1 generation, he began to see an interesting ratio in their offspring. He got a genotypic ratio of 1 to 2 to 1 and a phenotypic ratio of 3 to 1. This ratio is something that will be asked about on the test, so it's definitely a number that you should memorize. This is interesting and all, but Mendel wasn't satisfied, no. He decided to go and begin tracking how several different traits pass down through generations all at one time. For the sake of this video, let's just focus on the genes for the shape of a pea and its color. So now that we're dealing with 2 traits, the Punnett squares will look like this, a dihybrid cross. These are a little more complicated. You make these crosses first by finding the different allele combinations for each parent's gamete. Okay, let's break that down. Gametes are sperm and ova, the egg, and in this class we can assume that they will usually be haploid when dealing with Punnett squares. Haploid means that they have half as much genetic information as their parents. Instead of carrying 2 alleles for each gene, they'll only carry 1. In the context of Punnett squares, that means each gamete is only going to have 1 allele for each gene. Now, these combinations of alleles are not random. We get them by kind of foiling the alleles. So how I like to do it is first combine the first allele with both of the second gene's alleles, like this, to get your first two combinations. And then combine the second allele of the first gene with the second gene's alleles, like this. Of course, you have to do this with both parents. From here, it's the same as a monohybrid cross. You just go through and cross multiply. So when Gregor Mendel did this, he of course obtained the same results for the F1 generation. All of the phenotypes were the same. But for the F2 generation, he obtained a different ratio, and we can visualize it like this. 9 to 3 to 3 to 1. You should also memorize this ratio. Okay, other Punnett square questions. Sometimes you'll run into these problems that look really intimidating, but you'll just apply the multiplicative rule. Wow, that is a hard word to say. So instead of calculating all the genotypes at once, you just do them separately and then multiply the probability of your desired genotype together to get the probability of the whole genotype. Okay. This also applies to questions when you're asked like, oh, if I have 3 dice and roll them, what's the probability of me getting the number 2 3 times? I have a 1 in 6 chance of getting a 2 on a single dice, but if I want to get 2 on all 3 dice in a row, I would have to multiply 1/6 by itself 3 times. There are also some trick questions. So, for example, if you ever get a question like, Mary has 4 children and they're all boys, what is the probability that she will have another child that is a boy? The answer is 50% because in reality, your chance of getting a boy or a girl is always 50% no matter your past experiences. Also, the sum rule. Let's say if I have 3 chances to roll a 5 on a die, what is the probability that I get a 5? In this case, it would be 1/2 because instead of multiplying the probabilities together, you would add them. So you would add 1/6 plus 1/6 plus 1/6 to get 1/2. Okay, so let's get back to genetics. Let's talk about why alleles are inherited according to Punnett crosses. Meiosis. So before we get into this, I just want to preface that I will not be explaining the entire process of meiosis because there's millions of other videos that you can see and understand and learn. I also have a Quizlet on that, so maybe go study that and then come back. Okay, so after you've educated yourself, here are some things that you should know. People are diploid, meaning that they have 2 sets of chromosomes. In mitosis, chromosomes all line up at the metaphase plate like this and then split into 2 different cells. Mitosis is different from meiosis because cells do not arrange their chromosomes in the same way. In meiosis, instead of lining up like this, homologous chromosomes pair up and then split like this. They also go through meiosis too, where sister chromatids are further split up. This gives us a different distribution of chromosomes and explains how gametes are haploid. Another note about meiosis is crossing over. So during prophase 1, crossing over occurs. This is when homologous chromosomes, the pair of chromosomes that code for the same traits, even though they might have different alleles, exchange genetic information and swap DNA. This is obviously really important because it explains why human offspring are different from each other. Sure, they're a combination of your mom and dad's DNA, but you also have some further variation because of crossing over. Other important facts. Law of segregation and law of independent assortment. People always get these confused or are never entirely sure of what they mean, so let me dumb them down for you. Law of segregation is the fact that genetic information will be equally distributed to each gamete. So each gamete gets 1 allele for each gene, so that they'll have the right amount of genetic information to create a viable offspring. This is why we associate the law of segregation with anaphase 1 and 2, because that is when chromosomes are being allocated to each cell. The law of independent assortment states that no gene influences the inheritance of another gene when positioned on different chromosomes. We relate this law with metaphase 1 and 2 because that is when chromosomes are destined to different cells based on how they line up at the metaphase plate. Okay, chromosomal disorders. Some disorders that we discuss in class are a result of chromosomes not being shared or separated equally during meiosis. This is called non-disjunction. This occurs when spindle fibers fail or chromosomes do not line up properly during metaphase. For example, let's look at trisomy 21. So this disorder occurs when one gamete ends up with 2 chromosomes for the 21 chromosome instead of 1. When that pair of chromosomes forms a zygote with a properly formed gamete, you'll end up with a cell that has 3 chromosomes. Turner syndrome and Klinefelter syndrome are also results of chromosomal abnormality. An organism with more than 2 sets of chromosomes is a polyploid. Exceptions to Mendelian genetics: incomplete dominance or codominance. Some genes do not have an allele that is dominant to the other. In incomplete dominance, we see that in heterozygous individuals, the phenotype becomes a blend of the 2 versions of the trait. In codominance, both traits are present. Organisms will present with spots of each trait when it is codominant. Multiple alleles. Some traits have more than 2 alleles that determine the phenotype. Blood types is one example that we'd like to use in this class. In blood, there are 3 alleles, A, B, and O. A and B are dominant, while O is recessive. In AB individuals, the genes are codominant, so both types of antigens will be present in their cells. Sex-linked traits. These traits will only be passed down by people's X and Y chromosomes. Girls have 2 X chromosomes, so they have a chance to have the unaffected wild type of the gene or to be an unaffected carrier, or just to be affected. Males will always be affected by an X-linked trait because they only have 1 X chromosome. So it doesn't matter if a sex-linked chromosome is recessive or dominant, it will always be expressed. You'll see this when we get to pedigree analysis. If you see the word hemizygous, the problem is probably asking about sex-linked traits in men because it describes a condition where you only have 1 copy of the gene instead of 2. Linked genes occur when 2 genes are situated close together on the same chromosome. We call them linked because they are less likely to be separated during crossing over if they are close to each other. This means that linked genes violate the law of independent assortment. The only time they aren't inherited together is when crossing over occurs and they are separated. Polygenetic inheritance. This is the most complicated out of the list and will probably be on the test. This is when the expression of a trait is affected by multiple genes. It's kind of like the heavy metal tolerance in the plants that we looked at in the lab portion of this class. This is called the additive effect. One addition to this condition is that sometimes a certain gene may suppress or mask the expression of the other ones. In this case, the homozygous recessive genotype will always mask the phenotypes of brown and black. This is called epistasis. These polygenetic traits usually are continuous. Things like height, hair color, skin color. One telltale sign that a question is talking about epistasis is when the presence of one gene will always lead to the same phenotype, no matter what the other alleles are. Organellar inheritance. In humans, you inherited circular chromosomes in the mitochondria from your mother. In plants, they inherit this information in the chloroplast. Interpreting pedigrees. Pedigree analysis is how scientists study the inheritance of traits across generations in a family. The easiest way to learn how to interpret pedigrees is to understand the patterns that indicate the type of heritable traits. This image is pretty self-explanatory, but you have to memorize this for the test. These are the types of inheritance that you must be able to identify. Autosomal dominant / recessive. In these traits, both sons and daughters are affected. In dominant traits, they are present in every generation. Affected individuals have at least one affected parent. In the recessive version, it usually skips generations, and unaffected parents can produce an affected offspring. Let's look at an example. So we see that there are only 2 affected individuals. The trait skips 2 generations and affects one boy and one girl. The trait skipping 2 generations should be your clue that this is an autosomal recessive trait and not dominant. If you wanted to eliminate the other options, this is what I would do. It can't be mitochondrial because the mother of individual 1 in generation 4 is not affected. It couldn't be X-linked because the father isn't affected. XY linked. In these traits, you should be looking for patterns relating to how it is passed down to different genders. In this pedigree, we see that only males are affected. This may be an indicator that this trait is X-linked because it would be present more in males than females. It is also important to note that in the second generation, the gene is not passed from father to son, which means that this trait is not Y-linked. Let's visualize this. The father in the first generation is affected. This means that he has one X chromosome to pass on the trait. In the next generation, only carriers or unaffected offspring are produced. The boy is not given the carrier gene because his dad passing the Y chromosome to his son, and the daughters are carriers because even though they are guaranteed to get one affected chromosome, they are lucky enough to receive an unaffected chromosome from their mom. In the third generation, there are 2 affected individuals. We can substantiate the argument that this is an X-linked trait because their moms gave them an affected chromosome, which would guarantee the trait being passed down and displayed. Mitochondrial inheritance. This is the easiest to identify out of the 3. You are just looking for generations where all offspring are affected by an affected mother. In this example, we see exactly that. In the first generation, you see an affected mother, and in the second generation, we see all of that mother's offspring has the mitochondrial disorder. In the third generation, we see that only offspring that are descendants of affected mothers in the second generation are also affected by the mitochondrial disorder. And that's it. You made it. 16 minutes crash course. Good luck on the test. I will be getting an 100, so there will not be a curve. Sorry. Yeah. But anyways, have a good summer. Like and subscribe. And stay gaming, y'all. Stay on the grind. Hit me up on Steam. Yeah. Add me on Clash. Whatever. See y'all next year. And later.